right, hi everyone. Before we start off our talk, we'll give you guys a brief introduction about ourselves. So, as the slide suggests, my name is Vicky. I'm a current second-year student here in the Biological and Biomedical Sciences PhD program. I currently work in a lab over at Brigham and Women's, where I study Alzheimer's disease.
Great. My name's Noah. I'm also a second-year graduate student at Harvard. Currently, I'm working at a lab at the Dana-Farber studying cancer biology, but during my undergraduate, during college, I studied Parkinson's disease in worms, or modeling Parkinson's disease in worms. So I'm going to hand it over to Vicky to take into the first part.
All right, so the title of our talk is High Stake Stakes, the Science Behind Prions, Mad Cow, and Neurologic Disease. So as this talk is fairly meaty, we do have an outline. We will have an outline as soon as I figure out how to make the mouse work. All right, Plan B. All right, so our talk will be divided into 3 parts. We'll first off with an introduction into mad cow disease. Then we'll talk about prions and proteins, and then we'll finish off by covering other neurological diseases. All right, so part 1, mad cow disease, or how we learn not to eat the brain. So the year was 1985. And we are in the United Kingdom. What happens is an unsuspecting farmer finds out that one of his cows is acting particularly strange. He says his cow's kind of going a bit mad. Basically, the cow's not acting normally. Anytime he interacts with the cow kind of has a more bizarre reaction. The cow is very confused, and the cow is hard to control. So this started off as one case, but as time went on, we moved to 1992. And what happens is that 3 in every 1,000 cows in the United Kingdom has this set of symptoms. And all these cows are going crazy. And people call it mad cow disease because these cows are mad. So all this while this was happening, the British government was telling people, don't worry, this is totally okay. Though there may be something wrong with these cows, it shouldn't come to humans, it should just stay with cows and everything's okay. And then we get to 1995, in which the first human victim gets this mad cow disease and winds up dying. So this led to the question, what was making these cows go mad? And then also, what was making these humans go mad? So when we go and review the symptoms, what we see is that there's uncoordinated movement. Both the cows and the humans weren't able to move as well as they normally did. They were highly sensitive to normal stimulus. What this means is that they had a bigger reaction to what would be normal things. So for instance, if you turned on the light, they would shy away from the light a lot more than normal. If someone poked one of these cows or poked a person, they would also freak out more than normal. So basically, they were acting a bit more bizarre. They were reacting more to different things. They had unpredictable behavior. This one's pretty self-explanatory. You couldn't really tell what they were doing or what they were going to plan on doing. And finally, there was a general state of confusion, both in the cows and the humans. And all of these symptoms, when we think about them, kind of point to one bodily system. The nervous system, this case represented by a brain. So what exactly is the nervous system? The nervous system helps us interact with our environment. So the nervous system helps us take in cues that we get from our environment. So for instance, color, scent, location, vision, takes all this information and processes it, and then coordinates the actions of the body accordingly. So for instance, if you're just sitting in a classroom and a tiger just runs through the doorway, your eyes are going to see that tiger, your eyes are going to tell the brain there's a tiger in this room. And your brain's going to tell your body you should probably leave this room before the tiger gets to you. So that's generally what the brain does. It controls a lot of things, but that was just one example. So there are 3 general components of the nervous system. You have the brain, so in this cartoon over here, this would be the brain, so this is a human cartoon. You have the spinal cord, and then you have the nerves. This pointer makes it really hard to point out the nerves. But you get the general idea. So the brain itself is made up of a variety of cells. One of those cell type is known as a neuron. So the neurons send signals to each other and are very important in coordinating a lot of different functions and reactions. So at the bottom here we have a cartoon of a number of neurons. So for instance, if one neuron is trying to tell the other neurons, hey, we should do this, they'll keep sending that signal down the line so that all the neurons will relay this information downstream. However, as you can imagine, if one neuron were to die in this pathway, you're going to have a breakdown of this communication. So this neuron over here is being like, hey guys, we need to do this, but the neuron in the middle is dead. So the other ones are a little confused. They're like, what's going on? So generally what I'm trying to get at here is when there's a breakdown of this communication, when certain neurons die, you're going to lose a lot of functions in the brain. So going back to the idea that when they're looking at patients with mad cow disease and looking at these cows, they saw that they had a lot of symptoms that were related to the nervous system. So when they looked in the brain, they saw something that was a bit abnormal. So this image on the right, no, left, my right, your left, is a section of a human brain. So the purple dots are meant to be like the nuclei of the neurons, and the pink stuff is the other parts of the neuron. So that's normally what this is staining for. However, so here I'm pointing an example of like a nucleus of a neuron. But when we look at patients or cows that have mad cow disease, what we find is that we don't have the same type of staining. What you see is you have a lot of these kind of white holes in the middle, and those come as a result because the neurons have died and they've just left gaping spaces. So generally, you can see here there are purple dots for where the nuclei were, and the other pink parts are the general parts of the neurons, but there's still a lot of holes for where neurons used to be but have now died, so there's just empty spaces. All right, so mad cow disease is also an infectious disease. So what is an infectious disease? An infectious disease is caused when a foreign objects enter the body and damage the cells. So the reason why we knew this was an infectious disease because human patients and cows had similar symptoms. And when scientists dug a little deeper, what they found is that whenever these human patients had these symptoms and this disease, turns out they had had some beef from cows that had the disease. So it led them to believe that there was something being transmitted from the cows. So here's a cartoon of a sick cow. Something was going between the sick cow and the humans, but the question is we didn't really know what it was. So when we think of infectious diseases, or generally when scientists thought of infectious diseases, they know of two examples of things that cause infectious disease. And I'm sure you guys have heard of these. So for instance, E. coli is a bacteria, or rather, bacteria and viruses cause infectious disease. An example of a bacteria, I got ahead of myself, is E. coli. So some of you may have heard of this before. When E. coli gets into food, it can cause food poisoning. And then an example of a virus would be the flu virus, that thing that we're supposed to get vaccinated from every winter. And the general idea is that be it either food poisoning or the cold or the common flu, both of these are caused when infectious agents enter the human body and cause damage. So going off of that, in order for the scientists to figure out, what exactly was causing mad cow disease, they had to try to figure out what kind of infectious agent they were looking at. So if we think about how we normally kill bacteria, there are a number of ways. One way would be using a disinfectant agent. So a lot of you guys have seen Purell around. It's made of a high percentage of alcohol, so when you squirt Purell onto your hands and you rub it around, it's going to kill the bacteria. And another example is applying heat. So generally, we don't really eat raw meat, and one of the reasons is because there's a lot of harmful bacteria in the raw meat that would hurt us if we were to eat it. What we do instead is that if we apply heat and cook it, those harmful bacteria that are originally inside it are then going to die instead, and they're not going to hurt us. So here I have drawn these bacteria as these green cartoons. All right, so then when we go back to this idea of what exactly was causing the disease, was it a bacteria or a virus? So what happens was that when scientists actually took the meat from a cow that had mad cow disease and they cooked the meat, they found that the meat would still transmit disease. So when you apply heat, that doesn't kill whatever is causing the disease. So they ruled out that it would be bacteria because they knew that by removing the bacteria with heat, you still cause the disease, therefore it was not a bacteria. And so viruses have DNA, like a lot of the cells in our body. When scientists then treated the meat with different chemicals that would damage DNA and remove DNA, they found that it still caused the disease. Therefore, they ruled out viruses. It couldn't be viruses. So what exactly was causing the disease? So What it actually wound up being was a protein, which was very revolutionary for the field. People did not realize that proteins could cause disease. Specifically, it was a protein called a prion, which we've hinted at the title of our talk. So I'm just going to briefly define a prion here. So prion is an infectious protein that causes disease. The specific prion that is involved here in mad cow disease is known as prion protein, or is known as prion protein, or PRP. So Noah will go more in detail about prions and PRP and proteins in the next part of the talk. So we go back to our diagram here. Previously, when we knew we had a sick cow and something was going from the sick cows to the humans, before we didn't know what it was, now we know it's PRP. Now, the sad thing about mad cow disease and prions is that it's not a curable disease. So once someone gets the disease, there's nothing you can really do about it. So the best way to be able to prevent mad cow disease from spreading throughout a population is to find a way to kind of contain it, to kind of contain any diseased cows. So here we go. So this is basically comes down to like how the British government and different governments around the world dealt with mad cow to prevent it from harming more people. So here we have our cartoon again of our sick cow. So when there's a sick cow, the disease can either be transmitted to other cows or to humans. So the way that the cow can transmit the disease to other cows is that in the food industry, they used to do this practice where if there were ever parts of the cows that weren't used in the food industry, so for instance, bones or other bones or certain types of meat, they would grind up whatever is left over and feed it to other cows. I know it sounds terrible, they don't do that anymore. This is part of the reason why. And then when they did this, they would transmit the disease from one cow to other cows. And it's pretty straightforward how you get the disease from cows to humans. But this idea that, so if the disease transmits from a sick cow to other cows, those sick cows could also transmit the disease to humans. So in the end, the humans are really the ones that lose. So you see these arrows here. Generally, the government had to find a way to deal with these different arrows to prevent the disease from being transmitted from the cows, from a sick cow to other cows and to humans. So as I mentioned before, one way that they prevented the disease from spreading from cow to cow was to put a ban on feeding meat and bone meal to cattle. So basically getting rid of the practice of feeding parts of cows to other cows. I know that sounds really bad, because it was really bad, and they don't do it anymore, so that's really okay. And then the other way, the way to prevent infectious meat from going from cows to humans was that in slaughterhouses, they changed the way they process meat. So anything they considered so they changed their guidelines so that now anything that was nervous tissue, so brain, spinal cord, anything that contained nerves, were now considered risky material. So it's nothing that they would actually allow to be processed and be put into the food industry. So as a result, what we see here is, so this is a graph of mad cow disease cases throughout time. We have a spike here and about like in between, I think like 1992, 1993, about that time. And over time, the cases have gotten less and less. And this was as a result of changing the way that we process beef in the beef industry and making sure that no diseased meat got into the food chain and would harm other cows or humans. So that was the end of the first part of giving you guys a very quick overview of the history of mad cow disease, and here's a general summary. So first, mad cow disease is a neurologic disease. It affects the brain, and it's not caused by viruses or bacteria. Instead, it's caused by a protein, specifically PRP. Again, Noah will go more in detail into this in the next section. There is no cure for mad cow disease, but the way that we dealt with mad cow disease was by changing food processing to decrease the spread of infectious meat. So yeah, questions? Yes?
So yeah, you talked a bit about the nervous system. I had a question concerning how mad cow disease affects the physical or motor system that runs through our body. You talked about the odd coordination with hands and stuff.
So the question broadly was, how does mad cow disease affect the motor system? So as you can imagine, from the brain controls a lot of different systems, one of them being the motor system. There are neurons that go from the brain and then connect to our different muscles. And mad cow disease is a general kind of like dying of neurons all over the place. So what happens is that people and cows with mad cow disease do lose kind of control of their motor systems over time, because all of these connections from the brain to the muscles will disappear over time as those neurons die. Oh God, so many questions. I'm gonna go here.
How do we think it got into the cow population originally?
All right, so the question is, how did we think the mad cow disease started in the cows, generally? So there are a lot of theories. So one particular theory that I personally like is that there is a similar disease in sheep called scrapie. The reason they call it that is because the sheep, when they get this neurological disease, will go up to like fence boards and scrape themselves until they start bleeding. That's not something a sheep would normally do. It's sad. But they believe that somehow it jumped from sheep to cows. They don't know how, but as you can imagine, if there's even just kind of like exposed sheep blood from a diseased sheep that somehow got to a cow, it can cause the disease to happen. So that's one theory, but it hasn't been proven because people have been struggling to find a way to figure out how exactly it started in cows. Question.
Can it be eradicated?
Can it be eradicated? Not quite. So that comes down to, yes, I did repeat the question. So this comes down to like, can we really cure like mad cow disease? Because one way we could cure mad cow disease, if we can find a way to get rid of the protein entirely, we could then get rid of the disease, right? Unfortunately, that's not entirely possible. Part of it is because prion, this particular prion, PRP, is particularly strong. It has some characteristics that aren't normal for a protein. No, we'll go more in detail into this. So it's kind of hard to just develop something that would target this protein specifically and make it and completely eradicate it. So not quite. I'm gonna go. I think I saw your hand first. Yeah. So the question is, can cows be vaccinated? Unfortunately, not for prion disease, and this comes back again because it's a protein. So generally, the way that vaccines work is you kind of take a portion of a virus or a bacteria and you kind of train your immune system to recognize that, but prions are way too small for the immune system to be able to recognize. So there's no way, so you can't really take your immune system and train it to kind of fight against prion disease. Yes?
Accumulate in the brain of the cow. If you just eat meat, stay away from the nervous system, would meat still contain prion? Secondly, how does the prion get from the GI tract into the brain? It must go through blood.
Yeah. So the question is, how does the prion go from our GI tract into the brain, and follow up, if we were to eat other parts of the cow that weren't, the brain or the nervous tissue, would we still get disease? Generally, the answer is, so to answer the first question, we don't currently know how prions go from the GI tract to the brain. There are a lot of theories. It's an active area of research, but people don't really know how that happens. Now, the answer to your second question, if you eat other parts of the cow that aren't the brain or the nervous system, could you still get the disease? The answer is probably yes. But something I didn't mention is that while they did change the way that the way they slaughtered cows by removing the risky portions, so like removing nervous system, spinal cord, and neurons, they also, anytime a cow had mad cow disease, they're like, all right, we are not letting this cow enter the food chain at all. Like, we're just not going to process this cow. So that's one way that they made. So they just, even if a cow had mad cow disease, they wouldn't try to be like, all right, so this part is probably safe, and we can try to sell this to people. So the reason why, if you were to eat other parts of the cow that aren't the brain or the nervous tissue, could you still get the disease? There are some theories that prions may go to other parts. But this is getting to.
Does it get into the muscle?
It could. So could it get into muscle? Yes. I also do know that it does travel throughout the bloodstream. So that's when you get really, really difficult in terms of trying to really isolate out proteins. Question.
So I think it's said that sort of started to see increasing cases of mad cow in cows in like 1992-ish, and then it wasn't until 1995 we saw a lot. That we started to get cases in humans. presumably people were eating cows during that time. What do you think accounts for that sort of gap in transmission?
So the question was, there's like a kind of peak in cases in cows in about 1992, 1993, but we didn't see a human case until 1995. What accounted for this gap in time? So mad cow disease does take time to develop. So even though we had a peak in cases in 92 or 93, it would take a number of years before symptoms even start showing up in humans. And even by the time symptoms start showing up, the disease has already progressed pretty far along because a lot of neurons have died in the brain. So that generally is what accounts for it. So even now, today, like scientists are still actively monitoring certain places they believe had people that had eaten contaminated meat, because they're still trying to see if people are still developing symptoms, because it still could be possible, even though it's been, what's the math?
20 years?
20 years? Yeah, even though it's been about 20 years, people could still develop symptoms much later. But that would only be people that have been exposed to the initial meat, not anything after that, especially if with the changes in food processing and regulation. Question? Last question, but yeah.
I mentioned this is can you test, how do you test for the presence of prions, and is a test too expensive to just broadly apply to all slaughtered cattle?
So the question is, how would you, is there a test to test, or is there, oh wow, it's redundant. Is there a way to test for the presence of prion, and is it expensive? I don't know the answer to the top of my head. My best guess is that there isn't really a best way to test for it. Because the way that someone would have to do it would be to take the meat and to extract the protein, and then from the protein to be able to identify what all the proteins are. That inherently is a very expensive process in science itself, just in the process of doing research. So I can imagine it would not be something that they would be doing very often in cows. So there's not really a reliable way to do it. It's, or at least not a cheap or reliable enough way that people are doing it regularly as is, if that answers your question. Okay. All right, so I will now hand the reins to Noah, who's gonna give you guys some information about proteins and prions.
Great, thank you, Vicky. Great questions. So in this part of the talk, I'll talk more about what exactly is a prion, and maybe we'll hit on some of those questions again. Maybe it'll be more clear. And I'll also go over what is a prion. So as you just heard from Vicky, we learned the story of mad cow disease, and we learned that prions are infectious proteins. So in this part of the talk, I'll talk more about characteristics of prions in this top row. And in the third part of the talk, we'll talk about different other neurological diseases that have proteins involved that have characteristics like prions. But it will take me a bit to get to prions, so I just want to give you an outline of this section of the talk. First, I'll talk about proteins in general and how they're made. Then I'll talk about how proteins fold and get their shape. And we'll learn that prions arise from errors in folding, so I'll talk about those types of errors. So first, let's get into what is a protein. So many of you know proteins from foods, meat and fish have a lot of protein. I see there are a lot of bodybuilders in the room, obviously myself included. We know proteins as protein shake. But I want to convey that proteins are more than just food and more than just supplements for bodybuilders. They carry out a lot of cool functions in cells and in our bodies. So on the left here, we have this protein called an antibody that our immune system actually uses to detect viruses and bacteria and other things in our bodies that shouldn't be there. And what I want to impress upon you is this beautiful shape of the antibody. It looks almost like a Y, and these little bits out here are sort of like hands, and they can grab onto the foreign bodies. I want to also point out that the different colors actually represent different chains of proteins. So proteins have to sort of recognize each other in order to form these beautiful shapes. On the right is a protein called ATP synthase, which makes the energy of our cells, ATP. If you've heard of mitochondria as the powerhouse of the cell, it's because of the presence of this protein. And it's sort of hard to tell, but we think that this protein actually works almost like a turbine. So this part will spin and crank out ATP. Another wonderful protein in our body is hemoglobin. This is another computer-generated model of hemoglobin. And this aura here is sort of the surface of the protein, and inside these ribbon-like structures is the actual chain. And this rainbow-colored molecule is actually called heme, which is why we call it hemoglobin. And if you've heard of the Impossible Burger, this is the molecule that they started putting into to make it taste more blood-like. And hemoglobin is in our blood, and it's responsible for carrying oxygen, which are these blue spheres. So I know it's not incredibly intuitive right now, but I hope you'll take me on it that it's the precise location of these ribbons that allow the heme molecule to sit exactly where it's sitting and for it to transport oxygen. So the protein shape is really critical for its function. But what are proteins made of? So proteins are made of, as I mentioned, they're chains. They're chains of amino acids. And what makes them special are these extensions from the chains that we call variable groups, or R groups. And the different amino acids are defined by different variable groups. So I'll denote one type of amino acid, a gray amino acid, with a R1 type of R group, R2. And these different R groups have different chemical properties. So some of those properties are that they could be big or small, they can work as acids or bases, or they can have positive or negative charges. And so there are 20 different types of amino acids that go into building proteins, and they can form different sequences. And what the amazing thing is, if you think that there are 20 different amino acids, and that most proteins are made up of hundreds of amino acids, you could think of the sheer diversity of different types of chains you might get, which sort of explains how you get so many different functions of proteins. And so it might make you think, what determines the sequences of amino acids in proteins? And maybe you've come across it in earlier talks. It's DNA. So the DNA is actually the instruction manual for how to build these proteins from different sequences of amino acids. So now that we've talked about how proteins are chains of amino acids, how do you get such beautiful, intricate shapes? So again, remember that the different amino acids have unique chemical properties. And one important chemical property for a protein to get its shape is that some amino acids are greasy, and they don't like water. So on the left, we have a cup of oil and water, and we all know that they separate. So this property I'll define as being hydrophobic, from the Greek hydro, water, and phobic, fear or aversion. So a hydrophobic amino acid will want to repel water. So just to demonstrate that visually, again, we have two amino acids defined by different R groups. And I'll denote a hydrophobic amino acid with this orangey color to make you think of oil. And so if you have some water, the hydrophobic amino acid will want to repel it. And it turns out that the main driving force for proteins to fold or to take shape from an extended chain into a fold is they actually want to minimize the repulsion between the hydrophobic amino acids and water. So the hydrophobic amino acids will sort of clump up and try to separate from water. And if we go back to our friend hemoglobin, what I've done now is I've colored one half of the molecule. I've colored all of its hydrophobic amino acids in this orangey color, and I've left the other half as a reference. And what you can see is on the inside of the protein, you do have this hydrophobic core, a big group of hydrophobic amino acids. And so before I continue with my talk and get into errors of protein folding, I just want to take a quick pause for questions to make sure that this model is clear, that an extended chain of proteins, of amino acids that have hydrophobic amino acids will fold to try to repel water. Are there any questions about that? Great. So now that I've talked about what proteins are made of, I'll get into some of the errors of protein folding. So if you go back to our friend hemoglobin, you may have noticed that I cheated a little bit in directing your attention to here, and that there actually are some hydrophobic amino acids that do lie on the surface of proteins. So this brings us to the concept that protein folding is actually a dynamic process, and it's reversible. So you can have this extended chain of amino acids, try to adopt this one fold to minimize its repulsions to water. You can actually get the reverse happening and have this folded protein go back to the extended chain so that maybe it can adopt another type of fold. You can see a difference. And maybe this will have a better repulsion from water. And the stability of each fold depends on how hard it is to get back to this unfolded state to try to test out another shape. So this is a very dynamic process in cells. And what can happen is that you might get a fold that looks like this. That's not perfect, and you actually get an exposed region of hydrophobic amino acids that might be too stuck to unfold to get back into this extended chain. So what happens when that happens? We call it a misfolded protein, and the cell actually can adapt to this. The cell has machinery to try to unfold it and start over. The cells can recognize it and try to destroy it. Or you can get this third property of aggregation. I'll define aggregation as a sticking together of proteins, which I'll demonstrate in the next couple slides, or a clumping up of proteins. And I'll call one of these clumps of proteins an aggregate, which we'll use throughout the talk. So let me just show you what aggregation looks like. If you have two of these misfolded proteins with the exposed hydrophobic patch, you can sort of have them stick together like this. But I've called this innocent protein aggregation because it's just two. And actually, most of the cell machinery that I talked about in the last slide can recognize a small aggregate like this. So how does protein aggregation get out of control? Well, I'll just make a couple small changes, and I'll add another exposed hydrophobic patch here. So now when we have two proteins, they come together like before, but now you still have exposed hydrophobic amino acids. So more protein chains can come on, attach there, and again, now make things a little bit more complicated. These attach here, and bam. Now everything is totally out of control, and you have these large clumps of aggregates. And it turns out that these aggregates are super strong. So again, if we take just one of these small two proteins coming together, if you apply heat, you can actually separate them. But with these strong aggregates, if you apply heat, they stay together. So this might make you think of the meat from cows with mad cow disease, that even when you heat up the meat, you don't get rid of the prions, or the prion protein. So that gives us our second characteristic of prion, that they're misfolded and they aggregate. But what's the big deal? So what if we eat aggregates? So that will introduce us to our third concept, that prions can actually template the folding of other proteins. So templating is the process by which a misfolded protein causes a normally folded protein to then become misfolded. So I've demonstrated that here. Again, we have our extended chain going into this nice fold to try to repel water. And if we take this correctly folded protein and we add the prion aggregate, now this correctly folded protein becomes misfolded. So as it happens, PRP is actually, the prion protein found in mad cow disease, is actually found in the human brain as a correctly folded protein. We don't exactly understand its function, but it's there. And when the prions from the meat of mad cow disease interact with it, now we have it templating the misfolding of the prions in our brains. And you get these aggregates which eventually lead to neuron death. And so with that, we now have our three characteristics of prions. They're infectious proteins, they misfold and they aggregate, and they can also template the misfolding of other proteins. And so in the next part of the talk, we'll talk about two other neurological diseases where we see some of these characteristics. And to summarize this part, I've talked about proteins and how they're more than just food. They have a lot of different functions in our cells and bodies. Proteins are made of amino acids, and each amino acid has a distinct chemical property. And proteins fold to minimize the contact between the property of being hydrophobic, of repelling water. They want to minimize this repulsion. When proteins don't fold correctly, they can aggregate. And prions are aggregated protein that can cause other proteins to misfold. So I'll stop here for questions. Yes?
So why are the prions infectious? I don't understand that characteristic.
Great, so why are prions infectious is the question. So as Vicky defined earlier, infectious can be from the spread of cows to humans. So in humans, we have this prion protein in our brains. And normally we don't have the disease, but when we do when we find these prion aggregates from the meat of a mad cow, then it will misfold the proteins in our brains to cause disease. So now the prion from the meat now is causing disease, and in that way it's infectious.
How long has the word prion been with us, and who made it up?
That's a great question. So how long has the word prion been with us, and who made it up? Who got to name it? I'm actually not familiar with that. Vicky, do you know?
Come up to the microphone. So what's interesting was when I was doing some research into trying to find out more about mad cow disease, there were some articles printed in the late 1980s when they were trying to figure out what exactly was going on. And they're like, oh yeah, this one researcher in California, so keep in mind this is in the United Kingdom. So like some researcher in California in the United States thinks that a protein causes this disease, and he calls it a prion. And as you can imagine, there's a lot of skepticism. So it could be that he was the first person who coined the term, but I'm not quite sure if it existed before that. Katie looks like she's on Wikipedia.
A quick Google search has revealed that it was coined in 1982 by Stanley B. And it's a
Stanley
portmanteau
Prusiner,
of
yeah.
protein and infection.
Oh, great. So I'll just repeat it for the live stream, right?
Please do.
That a prion is from the term comes from 1982, and it's sort of a combination of protein and infection, so pre-in or prion, yeah. Okay.
So you talked about how PRP is found in the human brain naturally. Are there any theories at all as to what it could be used for? Because like you can imagine some memories not being needed anymore, and is that at all connected?
That's a great question. So the question is, what is the function of PRP in the brain? Do I have any theories? It also sort of goes into this idea of when the PRP from the cow affects this, is the disease caused from a loss of the function of this protein. So I don't think we totally understand its function. Some people think that's involved in transporting copper ions into nerves, so it might be involved in that type of signaling. But when you take mice and you remove the prion protein, we don't really see any clear phenotypes. So it is a current area of research. Anything to add?
An interesting thing to note is that the normally folded prion protein actually exists on the surface of the cells, so it makes it easier for misfolded prions to come in and cause them to misfold. It's not like the misfolded it's not like the PRP aggregate has to get into the cells, because the protein that wants to misfold is already out on the outside of the cell. So that's what we know about it. We don't really know too much.
Yeah, but that's a great question, and especially important for how we understand how disease is caused.
So this might be related to the function of PRP as it's found normally in the brain, but do we know how it causes neuronal death? How it kills the cell?
That's also an area of research, and we'll touch a little bit on that in the third part.
Right. Prions expressed in all types of tissues in humans, or mainly certain types of tissues?
That's a great question. So where do you find prion proteins? Is it expressed only in the brain, or is it expressed in all tissues? So it's not just in the brain, it's not expressed just in the brain, and it is an interesting curiosity why neurons seem to be specifically susceptible to disease. And you actually see that across different types of neurological diseases as well.
Make another interjection. So normal prion proteins also found in a lot of immune cells. So a lot of research believes that when the prion protein kind of leaves your GI tract, it enters into the immune system first. But because immune cells kind of turn over, so when they die and they kind of make more, but neurons in the brain, they don't do that. Once they die, they die. So that could be a reason why neurons are particularly susceptible to prions. That when they have the aggregates, they'll die because they can't recover from it.
Do they know the sequence of the amino acids in both the normal and the abnormal version of the prion?
So the question is, do we have the sequence of the protein, both in the normal and the abnormal state? So I'll also use this question to clarify that the sequence of the proteins actually is the same. It may have been unclear in the way I was trying to diagram it, but the correctly folded protein and the incorrectly folded protein is the same sequence. From humans to cows, it's slightly different. But yes, we do know the sequence of the proteins. It's just so the difference between the normal protein and the disease protein is just how it folds. Great question. Okay, so I think with that we'll take an intermission. Great, thank you. So before we move on to the third and last part, I do just want to clarify something that came up in the first question and answer session. I believe it was your question about vaccines for PRP and the use of antibodies. So it turns out that we actually can have antibodies bind to proteins, but the difficulty is, and then going back to your question, Top, is that the sequence is the same between the folded and the misfolded protein. So it actually is it's technically challenging to develop an antibody for the unfolded protein without hitting the correctly folded protein. But I will actually discuss that as a strategy later on in this part of the talk. So I just want to clarify that.
All right, so on to the third and final part of our talk. We're going to cover other neurological diseases and different approaches for treating these diseases. So going back to our idea of protein aggregation, so as we mentioned in mad cow disease, proteins aggregate and somehow they cause disease. So here we have our cartoon neurons again. These neurons will make a misfolded protein. I've represented this misfolded protein as a blue rectangle. These misfolded proteins will accumulate and aggregate together, and that aggregation will somehow cause neuronal death. Now it's important to note that as a field, we don't really know how protein aggregation causes neuron disease. It's a very, very, very active field of research in a number of different types of diseases, including mad cow disease. So yeah, that's why I boxed it and put like 4 question marks, because we really do not know what's going on. All right, so there are different, so if you think about this model of protein aggregation, the way that a lot of people have tried to think about therapeutics or different drugs to target this is by trying to figure out, a way to decrease the neuron making protein, and another approach is to decrease the chances of the misfolded proteins from aggregating together. So from this point on in the talk, Noah and I will introduce 2 other neurological diseases and how different therapeutics that work on this pathway that I've diagrammed here are being used to treat these diseases. So I'm going to start with Alzheimer's disease. I'm sure a number of you have heard this disease before. It affects over 5 million people in the US. The age of onset is usually about greater than 65 years of age, so it's a pretty late onset disease. So there's a lot of neurodegeneration in the brain, and one of the key symptoms is loss of memory. So what what happens is that neurons that are important for memory formation and memory retaining, memory formation and for retaining memory wind up dying. So here I'm not gonna make this mistake again. On your left, yes, I have a cartoon of a normal brain. So this is if you just take a brain and just cut it in half. That's what a normal brain looks like. When a patient has Alzheimer's disease, what we see is that the brain is much smaller than the normal brain. This comes as a result because a lot of neurons have died and the brain has lost a lot of mass. So we'll see these spaces here that weren't that big in the normal brain wind up much larger in the regular brain. Same with these holes here that were much smaller here. So overall, there's a lot of neurons that die in Alzheimer's disease. Now, one of the key accumulating proteins in Alzheimer's disease is known as amyloid beta. So here I have a picture of another type of staining for neurons. So all these blue specks over here, those are all neurons. And then these giant brown blobs, those are plaques of amyloid beta, so large accumulations of amyloid beta. And as you can see, they are much larger than the neurons. So they really do represent a substantial amount, so once these aggregates start forming, as you can imagine, they probably start to push neurons aside or somehow cause these neurons to die to really take up that much space. But again, we don't really know how amyloid beta and the aggregation of amyloid beta causes these disease. So let's go back to this chart that Noah had put together previously. So he talked about the 3 different characteristics of prion proteins. We have that they're infectious, that they misfold and aggregate, and that they template. So with amyloid beta and Alzheimer's disease, we don't really know if it's infectious. So this idea of if it can spread from one human to the other. I don't know if it's something people have been testing. I generally think no. And it does misfold and it does aggregate. And people do debate within the field whether or not a misfolded amyloid beta can cause other amyloid beta to misfold. So we just put a maybe here. So because amyloid beta doesn't have all three of these characteristics of a prion protein, we're going to call it prion-like. So it's like a prion protein because it has one of these characteristics. It misfolds and aggregates, but it is not a prion. So here we have our neuron. So I was talking previously about how one way we can try to prevent protein aggregation is to prevent the neurons from making these misfolded proteins. So here, if we zoom in on our neuron, we'll find something called beta secretase. So beta secretase is an enzyme. Beta secretase makes amyloid beta. So one therapeutic approach that scientists have taken is they generate these small things called inhibitors, these small molecules, and these inhibitors would bind to beta secretase and prevent beta secretase from making more amyloid beta. So this is something that currently is in active clinical trials. There's actually a clinical trial that just started this past December 2016. And it's currently now in phase 3. So basically what that means is they have a lot of patients that have Alzheimer's disease. They're giving them beta secretase inhibitors and seeing if it has any effect. So, oh yeah, so if we go back to our diagram about misfolded proteins and aggregation neuron death, if we prevent the neuron from making misfolded protein, we won't have misfolded protein. Therefore, there will be no protein to make aggregation, and with no aggregates that magically cause neuron death, the neurons will be happy and alive. So that's the general idea of how we can use inhibitors to prevent protein generation and to decrease protein aggregation. All right.
And so I'll discuss another strategy to inhibit aggregation leading to neuron death with the example of Parkinson's disease. So Parkinson's disease, 60,000 people are diagnosed with Parkinson's disease in the United States every year. Famously, Michael J. Fox has been diagnosed with Parkinson's disease, and he actually has an amazing foundation for Parkinson's disease research. There's a lot of resources there. It also talks about clinical trials. Parkinson's disease is a disease of aging. Only 4% of people that have Parkinson's disease were diagnosed before the age of 50. And the main symptoms of Parkinson's disease is a tremor, slow movement, trouble balancing, and stiffness of the arms or legs. And the key protein involved in Parkinson's disease, or that we see in Parkinson's disease, is called alpha-synuclein. And this is a blown up image of a neuron. So this blue part is the neuron, the main part of the neuron. And this big blob here, that's the aggregate of alpha-synuclein. So I hope you see from this slide and the slide that Vicky showed of amyloid beta aggregates that my slide of the aggregates getting out of control wasn't an exaggeration. They really are this massive. And so again, alpha-synuclein doesn't have all of the characteristics of prions. We don't know if it's infectious. It does misfold and aggregate. And whether templates or not, I think for alpha-synuclein, there's more of the field does think that it in fact templates. So if you have misfolded alpha-synuclein, it will cause normally folded alpha-synuclein in the cell to also misfold. So if we look at misfolded proteins, if we think about a strategy where you don't prevent the production of misfolded proteins, we can look at a strategy where we try to hit how the misfolded proteins start to come together to form the aggregates. So again, you have this blue misfolded rectangle protein as a rectangle coming together. But what if we use antibodies, which is why I wanted to make the clarification earlier. Again, antibodies are used by our immune system to detect foreign pathogens like bacteria and viruses. And again, you have these little arms here to grab on. So we can actually try to engineer the antibody to recognize the misfolded form of alpha-synuclein. And in that way, you take up the part, you bind to the part of the protein where another misfolded protein might come in, and you actually block that interaction from happening. And so in our larger diagram here, even if the cell is making misfolded protein, if we use engineered antibodies to block the misfolded proteins from coming together, we can block aggregation and hopefully block cell death. So I just want to show you some actual data from an experiment using these engineered proteins. So on the left, we have slides of brains. On the top, we have without any drug. Again, the blue dots are neurons, and these brown speckles are aggregates. And the stain used is SYN506, but it's not a critical detail. And then when you add the therapeutic antibody to mice that have this disease, you see a less amount of the brown speckles. And so on the right, we just have a graph actually counting that across all of the pictures, because one picture may be deceiving. And so when we chart the number of aggregates as a percentage of before treatment, so without any treatment, you have 100%. When you add the therapeutic antibody, you now get a 30% reduction, which may, at least on this graph, seem small, but we think that could actually make a difference in the course of disease. And it's currently in phase 1 trial in humans, which means that they're currently evaluating its safety.
All right, so the 2 drugs that we mentioned are currently in clinical trials. So the one on top, Verbustat, that was the beta secretase inhibitor that I mentioned previously, that's currently in phase 3 clinical trials. And then BIIB054, I know, a very appealing and attractive name, is the alpha-synuclein antibody that Noah had mentioned previously. An important thing to note that even though these are 2 drugs that are currently in clinical trials, they are 2 drugs of an extremely long list of drugs that are currently in clinical trials. All of these drugs in some way have to do with modulating protein production or protein aggregation. So as you can see, there's a lot of different directions that scientists are taking to approach these diseases and to try to treat them. So as a larger summary for this third part of our talk, so there are neurological diseases that have prion-like proteins that can aggregate. Again, these proteins are prion-like because they have some prion characteristics, but not all of them. And from what we have learned from prion diseases, by preventing and decreasing protein aggregation, we can somehow try to prevent disease. And there are many therapeutics currently in clinical trials. So there's a large conclusion for our whole talk. So PRP is prion that causes mad cow disease. Prions are infectious proteins that can misfold and aggregate, template, and cause disease. There are other neurological diseases that we've mentioned, like Alzheimer's and Parkinson's, that have proteins that are prion-like. And understanding how prions work and how these properties are unique to these types of proteins can help scientists understand and develop drug approaches for these diseases. So here is our thank you slide for Science in the News and all these amazing organizations. So yeah, we'll end on any last questions people may have. No questions? Yes.
The Parkinson is intracellular protein. And is it in the cytoplasm or in the nucleus? And secondly, how do you get antibody into the cell?
So that's a great question. So the question is, so alpha-synuclein.
What's the question?
Yeah, it's in the question.
Okay.
The question is, so alpha-synuclein is a protein that you find inside of cells, contrasted to amyloid beta that we talked about as being outside of cells. And so the question is, how do you get an antibody to actually detect the alpha-synuclein that's inside cells? Okay, that's good. So, and are the aggregates in the cytoplasm of the cells or in the nucleus of the cell? So to answer that question, they're in the cytoplasm of the cell. Alpha-synuclein, we think normally, before it's misfolded, acts to in vesicular trafficking, or a process of neurons to actually communicate signals between neurons. The question of how it spreads from neuron to neuron in the course of disease, and how an antibody can actually get at it during that, I think is still interesting and is an active area of research. Just for everyone else, antibodies can't go inside of cells, so there's sort of this challenge of how do you actually target something inside of cells. And so one idea might be that once the cell dies, then it can't shield the aggregate, so maybe that's how it's preventing. We're not sure. I'm not sure if there's a sort of an active mode to from one neuron to send the aggregate to another neuron, but that might also be a target for the antibody. That's a great question.
So we have alpha-synuclein, which is involved in sort of Parkinson's disease, which seems to have a lot of sort of movement manifestations as far as clinical manifestations go. And then you have the beta amyloid, which is sort of causing Alzheimer's disease, which has more like memory impairment, cognitive dysfunction. What do you think accounts? They're both plaque or they're both related to sort of aggregation of misfolded proteins. What accounts for the difference in like clinical manifestations of these two?
So the question is, so as we know, Parkinson's disease and Alzheimer's disease have different clinical manifestations. They have different symptoms, but they both involve the aggregation of proteins. So why is it that amyloid beta is specific to neurons that are involved with memory, and why is it that alpha-synuclein is specific to neurons that are involved in motor function? So that's a really good question. Actually, what's interesting is that when with patients with Alzheimer's and Parkinson's disease, you'll find that over time, as the disease gets worse, they start losing memory, losing motor functions. Now, it is it's an active field of research. People are trying to figure out why is it that with Parkinson's, it specifically targets one area of the brain, and why is it in, or rather, I guess the question is like, why is it that these diseases start off in a specific part of the brain and then spread to other places? That's something that we don't entirely know why at this moment.
One thought for Parkinson's disease is that you usually lose neurons that are involved in dopamine signaling. So there's a current section of the field that thinks that maybe the normal function of alpha-synuclein is to be involved in dopamine signaling. So maybe once that gets screwed up, then those specific neurons that are involved in dopamine signaling and the consequences, that's how you get that type of manifestation. It's a good question. Great, thank you very much.